All right, so this is part 4 of digestion, and I'm redoing this video, so I want to go back and touch on the concept of lacteals. So we had just got done talking about the small intestine, all right, and then there was this slide that showed that there were blood vessels that make it up to the lacteal. So there's an artery and a vein, and you can see the lymphatic vessel in green, and this particular lymphatic vessel has a name, and it is called a lacteal. And in these villi is where you're going to find these lacteals. And what they are going to do is they are going to absorb all of the dietary fat. That is how the fat is going to make its way into our body. It's going to enter the lymphatic system. And if you remember where the lymphatic system drains, it drains into the subclavian vein. So fats will circulate throughout the bloodstream. And some fats will then be stored in the liver for later use. All right, and then I talked about the histology already of the small intestine, so I'm going to pass through here. We went over the Brunner's glands, which were there to raise the pH in the duodenum. So as soon as the contents of the stomach leaves the stomach through the pyloric canal, these Brunner glands will then raise the pH so that that acidic chyme is no longer acidic. It'll become more basic, or another term for basic is alkaline, okay? And then the other part I wanted to touch on, so we went over the histology, and we were talking about that you have this sort of division here. This is where the stomach is, and here is where the intestine. So this is a great histological section, which has that the defining kind of mark between the stomach and the intestines. And you can immediately tell that you're in the duodenum because you can see the presence of Brunner's glands here. And you'll be able to make this out nicely under the microscope. And then another part that I was going to skip in the other lecture, but not for you guys, are another feature that you find only in the ileum. So the jejunum doesn't really have any defining features, okay? So the duodenum has the Brunner's glands. The jejunum is just going to look like your typical small intestine, like some of the features that you saw on the previous slide. But the ileum is going to have something called Peyer's patches. All right, Peyer's patches are found in the submucosa of the ileum. And this is part of the lymphatic system as well. All right, so another term for these Peyer's patches, so a more scientific term, is what is called mucosa aggregated lymphoid nodules. And we talked about lymphoid tissues in the lymphatic system, right? This has to do with the immune system and is going to help us mount a response if anything enters that tissue. So this long word that I just told you is abbreviated by MALT. That's what you'll see in the scientific literature. And what these Peyer's patches are doing is they are protecting the intestines from any bacteria that should not be in the small intestines. So they act kind of like tonsils would in the mouth. They're going to get rid of any type of bacteria or pathogen that should not be there. That is the role of the Peyer's patches. You will be able to look at the ileum under the microscope, a histology image of the ileum, and you will be able to see these beautiful Peyer's patches. Let me find a color that's going to highlight well here. Let's try yellow. Oh, that works nicely. So you can see all of these Peyer's patches here, right, in the submucosa. And again, these are little lymphoid nodules. They kind of act like tonsils. They are going to capture and destroy bacteria that should not be present there. All right, now I'm going to fast forward a couple slides because we already talked about the large intestine. All right, so I went over the anatomy of the large intestine, and I think where I left off was here in talking about the rectum and the anal canal. So that's where I'm going to start, and then we'll wrap up with the accessory organs. All right, so let's start on this slide here. All right, so the rectum is going to form like the last approximately 15 centimeters of the digestive tract. This is an expandable location, an expandable organ, and this is going to be for temporary storage of feces until the feces are removed. So we'll talk about the anal canal, which is that last portion of the rectum. There are some longitudinal folds there just so that it can expand and hold the feces. And then we'll talk about the anus. There's two sphincters that are located there. One of them is not under voluntary control, so this is under control of the nervous system. And then one of the sphincters we do have control over, and this allows us to pass feces when ready. So let me show you a diagram of this. I don't believe we have any models of this in the lab. So here is the anal canal. Okay, there is a rectal valve here, which will then move the feces into the canal when ready. You can see the rectum, okay? Is this anything kind of below the rectal valve? Let me change colors. All right, and then below that is the anal canal. And you can see that there are two sets of sphincters. You have the external anal sphincters here, here, and here. This is skeletal muscle. This is what we do control. We learn how to control this sphincter as toddlers. And then there's an internal anal sphincter, which is smooth muscle. This is under the control of the nervous system. There are a lot of anal sinuses here that are going to be releasing mucus into the area to help pass those feces. All right, so let's look at a cartoon of the histology, and then we will move on to a real image. So you can see the large intestine here and how a little section of the large intestine was taken and then blown up in this image for you to be able to see. The epithelium consists of simple columnar epithelial cells. And what you're going to notice on the histology image is that the large intestine has a tremendous amount of mucus cells. There's going to be a lot of mucus production to help move the feces along the large intestine. You're going to see a lot of intestinal crypts here. So we've lost all the villi now. We're moving into intestinal crypts. You can see the same layers that we've been seeing throughout the talk. The mucosa layer, the submucosa layer, the muscularis externa, which has 2 layers of muscles, and then the serosa or adventitia. And again, the sort of defining features are going to be those intestinal crypts and these mucus cells. Let me show you what the actual histology looks like. So this is a very typical image of the large intestine. You can immediately be able to identify this, or you should start to train your brain to identify this, because you see so many goblet cells here. That's what all of these cells right here are. These are goblet cells that are going to be secreting mucus. When we've entered the large intestine, we have lost all the villi. We don't need villi anymore in the large intestine because we are not absorbing any nutrients at this point. If anything, we're going to reabsorb some remaining water. These intestinal crypts that you see in the large intestines, they are sometimes referred to as the crypts of Lieberkühn. This was named after the person who identified these crypts. You may say intestinal crypts, though. I am completely fine with that. So if you want to use intestinal crypts and mucus cells, that works for me. So that's the histology of the large intestine. So that should kind of wrap up all. You should now have all the information you need to draw a diagram or a study guide to help you understand the different layers that are involved in the epithelium as you move through the digestive tract, and the key features and what's important structurally and functionally to make those changes. All right, and last, we're going to touch on, this is more of an FYI. You've probably heard of the microbiome, mostly because it's talked about all over the news, and there's all these products in the grocery store now that are targeting the microbiome. So just to give you a little bit of an idea of what the microbiome is, it has to do with all of the microbes, all of the bacteria that live in our gastrointestinal tract, mostly living in our large intestine. So this diagram is telling you the amount of bacteria that you can find all along the digestive tract. And what is striking is as soon as you hit like the distal end of the small intestine, you get a huge increase in the numbers and the types of bacteria that you have in your gut. And then look at what you see in the colon. So in fact, you actually have more bacterial cells living in your intestines than you actually have human cells in your body. That's the amount of bacteria or microbiome, the number of cells that we have that are living amongst us. And there are many different types of strains that live in the gut as well. And there have been a tremendous amount of research that has gone on in the past 5 or 10 years studying the microbiome. And what this research has told us is that actually the microbiome has a major impact on our health. And the types of bacteria that we have living in our gut can actually determine the types of diseases we have, our mental health, our skin health. It can affect our immune system. There has been studies linking the types of bacteria you have to intelligence, to diabetes. So really, everything you eat affects your microbiome, and you can actually change your microbiome by changing your diet. All right, I think that's about as much as I'm going to say. I actually worked on the microbiome for a few years, but I'm going to leave it at that. This is a nice cartoon image that shows you, I could only find images on Google for the stomach. I couldn't find all of the images, so obviously there's 3 regions to the small intestine, right? The duodenum, the jejunum, and the ileum. And I went on to tell you about some of the features histologically that happens in each of those areas. So make sure that you have taken notes on that. And then here's a cartoon image of the large intestine to kind of give you an idea on how to work through your assignment for next week. All right, so we are now going to move on to the accessory organs, and we are going to talk about the liver, the gallbladder, and the pancreas. All right, so we're going to start with the liver first. It is actually the largest gland in your body. If you remember it from the cadavers, it's about the size of a football. And it performs over 500 functions. There is so much that the liver does. But what we're going to do is we are going to focus on one function of the liver, and that is its role in digestion. So we are going to focus on bile production and how bile is produced in the liver. So the liver performs many metabolic functions. You'll probably learn a lot more about it in physio, but again, we are just going to focus on its role in digestion. So again, this slide is just highlighting some of those other functions that the liver plays. The liver plays a role in immunity. It creates blood. There's some fat storage and vitamins and other things that are being stored in the liver. But we're going to mainly focus here on this creation of bile. And I'll tell you how bile then goes on to break down fats and proteins. All right, so let's take a look at the anatomy of the liver. And this is a nice slide showing you how the liver is positioned in the body. So it's going to sit right below the diaphragm. You can imagine the diaphragm sitting right here of the thoracic cavity, and the liver is going to sit inferiorly to it. It's going to have a lobe that comes in contact with the stomach. So you can see the esophagus running here. You can see the stomach here on the side. You can see the duodenum right here. So let's focus on the liver and let's start talking about some of the anatomical features. So we'll first start by talking about the lobes. So here is an anterior view of the liver, and here is a posterior view of the liver. If you're looking at the liver in the anterior view, you're going to see two lobes. You're going to see the right lobe and the left lobe. Now, when you look at the posterior side, you're able to make out two additional lobes. So here you can still see the left lobe and you can see the right lobe. But what you'll also notice is that there's two smaller lobes here. The superior one is the caudate lobe, and the inferior one is the quadrate lobe. You can also see how the gallbladder is tucked in right there on the posterior side of the liver. All right, let's look at a couple other features. So this is looking at the posterior view. So the liver is going to have so there's a couple other, a little bit more complicated concept that we're going to talk about, because you can see that there is a hepatic vein and something called a hepatic portal vein. I'm going to go over the distinction between those two, but I just want you to make note of it right now. And then you can see the hepatic artery, okay? So we'll come back and talk about that. I just want you to see it right now. You can see the gallbladder also, and you can make note of the lobes. So I've highlighted the lobes in green, the veins in blue, the artery in red. We're going to work through the gallbladder next, so I'll tell you about the different ducts that make up the gallbladder. I think what I'll do is point your attention to the falciform ligament and the ligamentum teres. The falciform ligament is this ligament that's running right here that is separating the left lobe from the other lobes, okay? And then the ligamentum teres is a remnant of the umbilical cord during gestation. Specifically, the ligamentum teres is a remnant of the umbilical vein. That's not really something I want you to know. I just wanted you to know what that was. I won't ask that though. All right, let's keep moving. But do know what the falciform ligament does. All right, so let's take a closer look at what's going on in the liver and the types of cells that you will find in the liver. So here's a picture of the liver, and you can see this little square, this little microscopic section is being blown up here. And what you can see are these sort of wagon wheel patterns, all right, that you will find in the liver when you're looking at it under the microscope. This is actually the functional unit of the liver, and it is called a liver lobule. And I'm going to spend some time describing to you what this liver lobule does. So there's many thousands of these liver lobules within the liver. So let's take a closer look at that. So within the liver lobule, so this is a close-up of the liver lobule, you will find lots and lots of these cells right here, and they're lined up in like plates. These are hepatocytes. These are the cells of the liver. Hepato means liver. Hepato means, yes, hepatocytes. So these hepatocytes are lined up in the liver lobule, and they are going to be producing bile, okay? So you have these plates of hepatocytes that are going to be producing bile. Now, you can notice on the corner of these liver lobules, you have what's called a triad. And I think I have this, oh, here it is, a portal triad on each of the corners. And what you're going to find is a portal venule or a vein, a portal arteriole, right, an artery, very small vein, and a bile duct. That is what forms the portal triad. And right now I'm just focusing on producing bile, so I'm going to talk about the bile duct. So what's happening is that these hepatocytes are making bile. Bile is going to flow through these what's called sinusoids. That's what you see in blue here, these sinusoids, and the bile will be picked up by these bile canaliculi. The bile canaliculi will then dump into the bile duct. All right. So all along the liver lobule, these hepatocytes are making bile. Bile is going to dump into the canaliculi. It's going to dump into the bile duct, and then all of the bile ducts are going to merge together, and they're going to send bile to the gallbladder. Now again, I'm going to come back to these arteries and veins in a minute. I just want to get through this idea of the bile production. So let's take a closer look, looking at a couple different liver lobules. Again, you can notice the portal triad is going to be at all the corners. That's going to contain an artery, a vein, and a bile duct, okay? So again, these hepatocytes here are going to make bile. The bile is going to dump into those canaliculi and then dump into the bile ducts. All of those bile ducts will eventually merge together and head toward the gallbladder. And again, here's another view, a closer look where you can see the bile duct here. You can see the canaliculi right here. All these hepatocytes and all of these plates. You can see how the canaliculi is associated with those hepatocytes to pick up the bile that is being produced. All right, so as I mentioned, I was going to come back to what the arteries and veins are going to be doing. And so to kind of summarize this, and I hope to convey this in the simplest way possible, the liver is going to process nutrients from all of the digestive organs. So all of the digestive organs, all of that blood is going to drain into the liver because the hepatocytes are going to capture all of the nutrients, because it goes on to do a lot of things, but it's going to store all the nutrients from the food that we eat. All right, we can forget about this for right now. We already talked about bile. So This is kind of a repeat of what I was just talking about, but it's showing you images in histology. So you can see the liver lobule, okay? You can see that there's a triad on each of the corners. It's also depicted here, and you can kind of see how the bile is flowing in green. So again, the bile is it's going to go this way and head towards those bile ducts. All right, this is the other concept that I really want you to understand. So I've outlined it here, and hopefully it's very clear. All right, so we've talked about circulation, and we've talked about how all the organs, including the digestive organs, are receiving blood, right? So branches of the descending aorta are going to service all the digestive organs. You looked at this when you were looking at the torso model, when we were looking at the hepatic, the splenic, and the gastric artery, and the mesentery arteries, right? All the digestive organs are receiving blood, okay? All the blood that is leaving the organs of the digestive tract is going to enter the hepatic portal circulation, okay? So we need to pick up all the nutrients that we've just digested, and the liver is going to store it. So what essentially is happening is that the venous blood, so as the blood is drained from the digestive organs, as I've told you many times, the venous blood is oxygen poor. But when it runs through the digestive organs, it actually is nutrient rich. So yes, it's oxygen poor, but it's full of nutrients that it's picked up from all the food that we've eaten. And it's going to deliver that blood to the liver so that the liver can store all those nutrients. So what's going to happen is the liver cells, those hepatocytes, are going to extract those nutrients as blood flows through the liver. So let me show you this in a picture. So here is all of the digestive organs. All of the digestive organs are going to be receiving blood from the arteries, whether it's the splenic artery, the gastric artery, all of the arteries, right? And then through capillary beds, it's going to enter the venous system after it's been depleted of oxygen. But in addition to it, as it enters that venous system, it is going to pick up nutrients that have been absorbed, whether it was from the small intestine, the large intestine. Remember all of that absorption that occurs in the small intestine? That's going to be picked up. And so all of these organs are going to drain their blood. So let me stop for a second and say, normally in any tissue, the blood gets picked up in the venous system and it gets sent back to the heart. This is not the case for the digestive organs. The blood is actually going to make a pit stop at the liver before returning to the heart, because the liver needs to pick up all the nutrients from the digestive tract. So instead of going straight to the heart, it's going to go to the liver first, okay? So it's actually spelled out nicely right here. Most of the blood from the abdominal organs goes into the hepatic portal vein first instead of the inferior vena cava, because the nutrient-rich blood from the digestive organs is going to go to the hepatocytes so that the hepatocytes can grab all the nutrients through the portal system. And then after processing the blood, so after the nutrients are taken out, then the blood will head back toward the heart through the hepatic vein. So this is actually a great diagram explaining how that is occurring. So again, if we look at this, so here is that portal vein. All of the blood from the digestive organs is going to enter the portal vein first. It's going to flow up through here, and then it's going to run down those sinusoids. And those hepatocytes line the sinusoids. So as blood is flowing, the hepatocytes can grab onto all the nutrients and store it. And then the blood will then be sent up here through this central vein, and then it's going to go to the hepatic vein, which will then make its way to the inferior vena cava. Please also look at your book, and if you have any further questions, I am happy to answer them when we are in lab, and hopefully you can piece all of this together. All right, let's move on to the gallbladder, and then we'll hit the pancreas and we will be done. All right, so the gallbladder is going to store and concentrate all of the bile that is made by the hepatocytes. And it is going to expel this bile into the duodenum. And this is what is going to help emulsify or break down fats that we've eaten. So let's look at the anatomy of the gallbladder. All right, it's a pretty simple anatomy. The only real trick is memorizing the different names of the ducts. All right, so imagine that the liver is up here, okay? And so as the liver is producing bile, it's going to flow down what is referred to as the common hepatic duct because it is from the liver. All right, and then if it's going to be stored in the gallbladder, the bile will then transfer or be sent down the cystic duct. The cystic duct is what leads to the bile duct, or to the gallbladder. I'm sorry. All right, and that's where bile can be stored. When bile is needed after we've eaten something that might have some fat to it, it will leave the cystic duct and then enter what's called the common bile duct. And this common bile duct is going to head to the duodenum. All right, so the gallbladder has a pretty simple anatomy. It has a body, a neck, and a fundus. Remember a fundus from the stomach? It's like a pouch-like feature. That's the anatomy to the gallbladder. It's pretty simple. It's just memorizing the different ducts and knowing how bile flows. All right, so here's another image, but now we're putting the gallbladder next to the duodenum so you can kind of see how it can release its contents into the duodenum. So you see the gallbladder here. Some of the ducts are labeled here. Here's the common hepatic duct, so that means that the liver is up here. The cystic duct is the duct that leads directly to the gallbladder. And then you have the bile duct right here, which is going to release its contents into the duodenum, okay? And this is going to, again, break down fat. There's a little sphincter right here, okay, because we want to be able to regulate when and how we release bile. So it's called the hepatopancreatic ampulla or sphincter. Either one is fine. And the reason it has this name is because it is in close association with the pancreas. So you can see the pancreas right here. It is tucked in to that C shape of the duodenum, and it is going to share, this sphincter is shared both by the bile duct as well as the main pancreatic duct. And I'll talk about what the pancreas is doing in a minute, but the pancreas is also going to be releasing juices, and they both will enter the duodenum at the same location. All right, this next slide is outlining the names of the ducts that I already went over. So in case you need it here, it's all written out here. I've already gone over it, so I'm probably not going to read it again. But if you need the information of the names of the ducts or where the bile is flowing, it is listed here. All right, the last topic we'll talk about is the pancreas. All right, and the pancreas actually has dual functions. So it has two specific roles. One of them is an exocrine function, and this has to do with digestion. One of them is an endocrine function, which is formation of hormones. I'm going to give you the very basics on this particular role. You will learn more about it in physio, but I want you to at least understand that the pancreas does have two roles, and please know what those two roles are. So first, it has a role in digestion. There are cells in the pancreas called acinar cells, and they are going to make, store, and secrete pancreatic enzymes. All right, they are alkaline or basic. They are going to help to raise the pH, because remember, the pancreas is also going to dump into the duodenum. And again, we're trying to raise the pH from that very acidic chyme that came out of the stomach. So the pancreas is going to release these enzymes that are in a very basic solution. So these enzymes are going to be activated in the duodenum as soon as the pH raises. And these enzymes are going to digest proteins, carbohydrates, and lipids. That's the role of the pancreas. It also has another role. Again, it's an endocrine role, and it's involved in the production of insulin and glucagon. And this is what is going to regulate our blood sugar levels. All right, so the pancreas also has a very simple anatomy to it. There is a body, a tail, and a head. Okay, that's the anatomy. That's all I need you to know of the pancreas. But what's important is this main pancreatic duct. That is the duct that all of those pancreatic enzymes and juices are going to flow through to reach the duodenum. It's going to meet up with the bile duct. And again, they both enter at the same location. There's a little sphincter there. It's also called the duodenal papilla. Okay, it's like a little nipple-like projection that is going to release those, both the bile and those pancreatic enzymes. Let me show you a cartoon image of the histology, because we're also going to look at this under the microscope. It's really interesting to see. So let's first talk about let's start with the exocrine cells. So again, exocrine has to do with digestion. So if you take a closer look at the pancreas right here, you can see that, so here's that main pancreatic duct. There are clusters of cells called acini, okay? Within the acini are these individual acinar cells. And these are the cells that are going to be producing those pancreatic enzymes that are going to be needed for digestion. So when you look at the histology, the pattern that you see is you're going to see those acini, so these clusters of acinar cells. So there's clusters of them. And then in the center, right here, is where you get that endocrine production. So there are these cells called alpha cells and beta cells. Let me just, again, it's just a brief introduction of what they are. So the alpha cells are the ones that are producing glucagon, and the beta cells are the ones that are producing insulin. And this is going to regulate how much glucose is in our blood at all times. And the cluster of these two cells together form what's called, and again, this has to do with the person that identified it, they're called islets of Langerhans. Islets of Langerhans, okay? So what you will see are the acinar cells here on the outside, and in the middle are the islet of Langerhans. So that's what you're seeing here in the middle. They're like islands, little islands, okay? Islets of Langerhans. And there, again, they have to do with blood glucose level. But that's really the extent to which I need you to know. So this is what the actual histology is going to look like when you look at it under the microscope. So again, you will notice lots and lots of these acinar cells. These are exocrine cells. And then occasionally you'll find the islet of Langerhans. So this is what it'll look like under the microscope. Remember, the pancreas has two functions, exocrine and endocrine. I think I have one more histology slide. All right, so here are those. So you can see clusters of those acinar cells. All right, I'm just kind of making up circles here, but you get the idea. And then in the middle is that islet of Langerhans. All right, so I think hopefully you have a better understanding of what is going on in digestion. And I think that wraps up our digestion lecture.